All right, that brings us to the top of the hour. Again, my name is Robin Komucheka. I'm a technical application specialist with de novo software and Dotmatics. And I'm really excited to be hosting this webinar today on getting started with FCS Express 7 image cytometry. If you can hear my voice and see my screen, please do us a favor and just say hello in the chat. If you have any questions throughout the webinar, please ask your questions as we go. My colleague Greg Veltri is joining us today and he will be answering your questions in the chat window. If it is best for me to provide a demonstration to answer your questions, I'll do that as we go as well. I really want you to get the most out of today's webinar, so if you do have questions, please, please, please don't hesitate to put them in the chat. If you find you have questions after today's webinar, please reach out to us at support@denovosoftware.com. Don't worry, you will hear and see this email address again later in the presentation. FCS Express is not only a flow cytometry analysis software, but it can also provide the tools you need to analyze your image cytometry, microscopy images, and other imaging platform data. Today's webinar is going to be a great start on exploring what FCS Express has to offer by introducing you to some of the tools, features, and capabilities. I'm going to give you a little bit of history to get us started regarding image cytometry analysis with FCS Express. We actually launched our first image cytometry analysis and presentation software package in version 4. It was previously a separate version of FCS Express, but since version 7.18.0015, we've brought those tools together, so now FCS Express contains these tools for working with image cytometry by default. The team at de novo software and Dotmatics is always continuing to make improvements to FCS Express, and we are recently included zooming and panning options for picture plots, and we're always increasing the image cytometry data file compatibility. So to get started, we're going to start with just like the workflow. It's a little bit different than we're coming from flow cytometry. First, we have to acquire those images. Second is image segmentation. Now, this is not always necessary for all image cytometry data files, but some you will need to have a go-between to get those files ready to be brought into FCS Express. So what is segmentation? Well, segmentation helps us define cell boundaries and populations of interest, and it creates distinct objects. After we've done that part, we can take those distinct objects and we get to extract features that provide additional information such as light intensities, shapes, sizes, and other identifying properties that can be used as parameters. FCS Express can handle as many features or parameters as your third-party software can provide. For segmentation, there are both free and paid-for varieties, and we can read from a variety of different file types from the image cytometer as well as from these segmentation tools. So let's get into compatibility a little bit more deeply. You can see here we have quite a great list of what we are compatible with: AMNIS, DAF, or FCS files, CP out files from Cell Profiler, that is a segmentation tool which is free, ACS files, ICE files, Life Technologies Tally data folders, ISO files, Nexstat files from Nexalom, as well as ICE files, IJ out. ImageJ is also a segmentation tool. We have Perkin Elmer, Vectra, TIFF or text files. We have ICE files from multiple different vendors. And then we have ACS or FCS files from the Attune site picks. I do have a quick note for you here. Image to gate linking for Vectra Inform datasets is only currently available for exports from Inform software releases prior to Inform 2.4.1. Raw data import analysis is still available, and we are planning in the future to have that compatibility again for image and gate linking. Perker Elman Acapella folder is available in FCS Express 7, but it's only in the legacy data file formats, and the Columbus database link was depreciated with FCS Express 7. If you do have any questions about your compatibility with FCS Express, you can always contact us at support@denovosoftware.com. Now, moving into analysis, the great thing about analysis and the tools that we have is there are so many tools that overlap between flow cytometry and image flow cytometry. So we use those and really exploit those to have those plots and additional features and tools to build an analysis strategy that really gives you the most from your data. And then we use those images to confirm all the other findings that we have from those plots and histograms. And then we can do report generation. So once we have established a analysis strategy, we can take that information, report it out to PowerPoints, PDF, Excels, many different ways and options to get this information into reportables outside of FCS Express. So as you're actually building your analysis strategy, you're actually building those exports as well. As you can see today, I am actually presenting from FCS Express, and that can be really useful too, because if you need to do a short little meeting with your lab, you can just present from your FCS Express, so then you don't actually have to create any exportables either. So the topics that we're going to be covering today, like I said, this is a getting started. So we're going to get familiar with the interface, the tabs, where things are, where to find things in FCS Express. We're going to be using those picture plots, using the data list, getting familiar with how we move data, how we bring data in, gating, looking at our gating tools to see how gates are applicable, move them around a bit, some data grids, statistic tables and text tables, overlays, batch processing, and saving this layout. One thing that's amazing about FCS Express is once I have created a workspace or a layout, we can create it as a template. So all I have to do is bring in new data, reuse old plots and things. It will update with the new data, move things around, and report them out. So it really can be efficient and effective way to do your analysis. Now let's move into some analysis. We're going to start with our CP out data, which again is a segmentation data created from segmentation. You'll see here that on my side tab, I have actually tabbied some of my references, frequently used things that I use in FCX Express. So here's my data list. I had it shrunken to the side. If I click this pin, it will pop back out. And then I'm going to do the same for my gate navigator, just so we can look at some gates. Now I was working in this a little bit earlier, so you see I have some gates already established. If you have gates, you can always just delete them out, and that's what we're going to do quickly now. And if it has dependents, it will give you that little window to ask and make sure that you want to delete this, and letting you know that every other gate will be deleted as well. Let's get familiar a little bit more with the space too. On the File tab, we're going to have all of our tools to open, save, get some info about our software, and then most importantly, we have this feature called Options. FCS Express User Options has a lot of different options that we can select for our image data. So you'll see here down at the bottom, we have different image properties folders for vendors that we can set some properties of how we're going to load this data. These can be really important to set. You always want to make sure that we're loading images. We can do some things to optimize how data is coming into the software for different data file formats. We also have a ton of different other user information and options here. This really, what it does is it customizes this workspace. It will keep this information in a file. So then once you have set up your best, most optimized settings for your analysis, the software is always going to remember that and bring it in for you. So we're going to go ahead and cancel out of this window. On our Home tab, we have some features like arranging and aligning, which is really nice because again, as we're building this analysis strategy, we're also building our exportables to PowerPoint and PDFs and things. So we want to be able to arrange that nicely, neatly, and have the ability to customize what our plots are looking like. We can add new pages, delete new pages. We have some texting, text font and formatting we can do. And then we have some tools to paste and copy. You'll see that this is a lot like PowerPoint. We did have the similarity so that if you haven't been in FCS Express before, you will have some overlap and be able to find things a little bit more easy navigating this space. On our Insert tab, you'll see some shapes and text box, the gate view, which is another way to look at our gates and how they're assorted or in hierarchy in relationship with another. We have pictures, data grids. Here's our plots that we can introduce, and then we can do spreadsheets and charts, which is taking your analysis a step further without ever having to leave FCS Express. I can do a lot of those advanced statistical analyses and also introduce charts to really represent my data well. On the Design tab, we get to tell the software how we want to use our pages. Do we like portrait? Do we like landscape? I'm a landscape person. But then we can also define margins. Do we want to create margins here? And then some layout ideas if we are just trying to organize how do we want to set up this layout to get started with our data analysis strategy. On our gating tab, we'll see we have plenty of different gates. One thing that is newer to FCS Express is our quadrants are our gates by default now. You don't have to convert them. In previous versions before 720, you did have to convert our quadrants to gates, but now they are defaulted to gate behavior. Our marker, which we'll get into a little bit later, does still require to be converted to a gate, which is a great tool because sometimes we just want to mark some information, pull out some statistical information, but we might not want to commit to a gate. Batch and export. This is how we get all of the wonderful things we do in FCS Express out into reports. We're going to go into this a little bit deeper, more deeply towards the end, but we are able to add quite a few different actions telling the software what reportables we want. If we want to print a layout, save a layout, report as a PowerPoint or a PDF, we have exports to Luma and Prism. So we have quite a versatility to make sure that you're getting the results you need to the places you need so that you have all the results that you need at the end of your analysis. On our text tab, we're going to control all the fonts and things, and we also have some tools because we can insert tables into our text boxes. Data tab just helps us organize and arrange our data, make sure that we're optimizing the use of data, moving it when needed, and then adding some different tools like viewing header, which gives us some information about what's contained within a data file. On our Tools tab, we do have concentration calculator, channel calibration, and different tools to really help if we need to get more information. We need to know how many cells per mil. We can extract that information. We also have some standard operating procedures. On the Quality tab, we can run some alerts, make sure that we aren't missing anything. If we're sharing this layout or analysis with somebody else, we can make sure that we're also performing things in the same way. On our View tab, we have different things that we can add. Custom tokens are a biggie. Tokens are really just pieces of information. So we can customize those to gather the information that we need and bring it into our workspace. Wonderful things about tokens is once we've inserted, as we move through data, they update automatically with new data files, so we don't have to create them again. And for their navigators, we have a layout navigator, which is like a table of contents, tells us all the things that are happening on each of the pages of our workspace. We have a data navigator, which is going to tell you where your data files are being used, and a gate navigator is telling you which plots are using these gates, and it's open down below. So this is now default open when you open a workspace. And then view. So if you want to view a couple pages at the same time to just look at your analysis, that can be really helpful. On our help screen, we do have information about linking to different resources and then about your license. Because when you write into support, we'll often ask you, what version of FCS Express are you using? If you click on that, you'll see that version number right up there at the top. And then we can get some information about your license and make sure that we're giving you the proper guidance. All right, without further ado, let's go ahead and jump into adding our data. So with all these different data formats, it is going to be really important that we select the right format when we're looking at different data sets. So this first page is going to be for our CP out or a cell profiler segmented data. And right now my format is set to ICE, so I'm not going to see the data file. But as soon as I switch it back to the correct format, I see that I have 4 data files here. I can select one, I can select multiple and bring them into the software. And then when I click open, they'll appear in my data list. You can also drag and drop. You can drag from the folder onto the workspace. So there's many different ways to get started in bringing in your data here. After I've got my data populated in my data list, I'm just going to drag this file out. When I drag this file out, I can select different plot types. So here I'm going to go ahead and do just a color dot, and I'm going to go do a picture plot as well. And you can select as many or as few from this window. And once you click OK, they're going to appear in your workspace. So starting with our picture plot, I'm going to go ahead and right click to maximize this just so you can see it in all its full glory. We have a cytoplasm channel and then we have a DNA channel. So you can see with the same image, we actually have 2 views, 2 ways to view these cells. And then if when you're done maximizing, if you right click, you can click restore and it'll pop back to the size that it was. In our color dot plot, right now we don't have any color because I haven't created any gates yet. But on these parameters, if you click in the center, you'll be able to get the list of parameters. If you see here, we have an extensive long list of parameters. If I click on my data tab and go to that view header, we get all the information that is stored within this data file. And you'll see I actually have 74 parameters. Now that can be a lot to manage if I have to go through them one by one. So one of the beautiful things that we can do is we can start typing and it will reduce the number of parameters that are showing. So AERE is not very specific, so it didn't reduce that quite so much. But once I find the parameter, I can click on that there. For the next one, I'm going to look at some intensity parameters, and we're going to start with cytoplasm. Now, if I look at these dots, they look a little faint. It can be hard to tell where my events are and where they're not. So one of the tools that we have is we can format this plot. So if I go to overlays here, you'll see my format window automatically docks to the side of my page. I can change that. If I wanted to drag this out, I get these little plus icons and I can tell it to go to different sections if needed. I'm just going to kind of return it to where it was back over on the side here. And then I can just continue on. It just is a way to give yourself space. Also, these pins, if I click on a pin, it will hide over to the tab there so then it doesn't take up space as I'm working through my analysis. For now, I'm going to repin it back to being open. Here I'm going to increase the dot size because I really want to make sure that I'm selecting populations, I'm not making anything up, and I'm seeing actual events as I start gating. From the gating tab, I have all these selections that I can make to choose the gate that's appropriate. I'm going to start with this rectangular gate, and I'm just going to create it here in the middle. Whenever I create new gate, I do get this new gate window. I can name this whatever is going to be most fitting for my populations. I'm going to do mid-bright cyto because I think we're looking at the intensity of the cytoplasm channel right now. I can choose a color. And then if you expand the bottom here, you can choose to do different things with this gate. So I can redefine a parent gate if there were gates already established. I can emphasize events on color dot plots, which will be really useful in just a little bit here. I'm going to show you why. I can also create data-specific gates when moving, which means that this gate will be in a specific location for each of my data files. So it doesn't have to be a universal placement. FCS Express is a little bit different in that we start with a universal placement and then we define it to be specific after. And we can also exclude events. So if I need to create an exclusion event, I would just check this block. I'm going to go ahead and leave it at default and then just click OK. Whenever I create a new gate, we do get this little tip window the first time. It just tells you that you can drag this gate out to create a new plot, which is super useful, but after you've seen it once or twice, you may not need it anymore. If you just check this box to do not show this tip again, and then click OK, this window will go away. Everything in FCS Express is formatable just about, so if it's something you want to see in a specific way, we're definitely going to have the tools and capabilities, or most likely going to have the tools and capabilities to make that happen for you. As you can see, as soon as my gate has now been applied, I can see different populations here in my plot. Now, if I move this around, I'm going to see that that is actually updating in real time. So that's one of the wonderful things that FCS Express also does is we have the opportunity to update with all of our gates. So if you create a gate and then you have statistics reporting or other plots depending on this gate, everything downline is going to update in real time. I should also introduce at this time with this picture plot, we do have the option to do some zooming and panning. So we can do some zooming on this image and I can just select that zoom option, select a region, and then it will zoom into that plot. If I want to go to specific options, I can also change how this zoom is. So as you can see, I'm kind of looking just at this upper left-hand corner of this plot. If I want to start looking around at other regions of the plot, I can change by just dragging this little red square around. I can also zoom out so that I'm not just looking at things so closely. So I can change the zoom so that I'm getting the full field of view. and have a little bit clearer depiction of my sample there. Now for the next, I'm just going to drag this data file out again, and this time I'm just going to do a color dot, click OK, change this to shape, oops, area, and then also back to intensity, but this time I'm going to do it for the DNA plot. Again, our shape here is the dots here are a little undersized, so I'm going to increase them. As you see, as soon as I introduce this, the events within that initial gate are already showing on the plot. So I already have my mid-bright population there. On this plot, I'm going to introduce a new gate. And this one is just going to be my bright DNA. And click OK. And now you'll see that I have my red cells here. I also have the blue cells, and as I move these around, this is also updating. And you're not limited to just making gates on the 2D plots or the 2-parameter plots. I can also create a gate here. So I'm going to right click to maximize this plot again. Let me undo that really quickly. There we go. And then I'm going to introduce just a polygon gate. And now I'm going to select kind of a weird population, just some of the cells that are not falling into either of those gates as of yet. And then I'm going to actually adjust these gate properties so that they are emphasized on color dot plots. I really want to make sure that they're larger so that I can see them well because I'm looking at very few events. And then I'll click OK here. I'm going to right click to restore. And then I have my image here. So it kind of changed my panning a little bit. So I'm going to go back to the specific options and just change my zoom a little bit so I can see that gate. The gate here is in the middle. I don't know if you can see that very well. It's kind of hidden. So one of the things that we can do in gate properties as well is we can increase that line width so we can make it really clear where a gate is falling. Also, the color is probably not great for all the colors that are there. So I'm going to make it a bright green, and then I'm going to click OK. So now I can see I've got those cell populations, the line is very visible, and then those events are very bright as well. And if I were to move this around, again, you would see those different cells for a different population. As I go to the second data file, again, my zoom is a little bit different here, so I'm going to go to my specific options and just change that zoom so I can see that gate. If I need to adjust this for this data specific, to make this data specific for this file, I can take this population, click on the data specific gate, and then I can move it to a new location. So it's going to be here for that second sample and there for that first one. So I can have different positions for my gate as needed. Now, with statistics, because statistics are super important, we want to know information, get this information out of the software. If I right click on any plot, I can right click and show gate statistics. I'm going to just pull this over here. You'll notice that even if I get my statistic table gets separated from the plot, when I grab it or highlight it, it automatically grabs the plot. This is really important because FCS Express approaches statistics on a plot by plot basis. And what that means is the statistics shown will be relative to the gate that's being displayed on the plot. So right now I have no gate, but if I were to change this using the drop down in the middle here, maybe do bright, you can see that my statistics also had changed here. You can also change the gate that's being displayed from the gating tab, but it's also nice and convenient to just go ahead and change it right here from the plot itself. Now with this statistic table, you are not stuck with the statistics that we display by default. If you want to change the statistics here, make sure that you're grabbing your statistic table, and then you'll see we have a statistics option. We can start deleting or adjusting which information we're showing. Maybe leave X and get rid of that. And then we can also say which gates to display. So if I had a huge gating hierarchy and I didn't want to show them all, I can show no gates. I can show only the gates checked below. So it will wait for my input to tell it what gates I'm interested in showing. Or I can do all gates that are checked below. Oh, sorry, all gates except those checked below. So we'll just return to all gates right now because we don't have a lot of gates as we're just starting to get into this analysis space. Now I want to move us to another data file type. So in my data list, I'm going to add again. It will always bring you back to the folder that you were most recently working in when you're working to add data or things. Again, this file type is going to be really important, so you just want to make sure that you're grabbing the right file type so that you can see the files that you're interested in. And this one, I'm going to click, I'm going to select all three and click Open File. Now this is data file from the Cytometry, and here I'm going to introduce a color dot plot. That should be fine. And what's really interesting with the site picks is that there are some features that we can evaluate whether an event has been processed. Oh, let me go here. Right. Good. There we go. Change the parameter to event there. Now we can see that the event has in fact been processed. And then we can also duplicate this plot. And on the second plot, I'm going to actually look at the particle count. So one thing that's great is that the instrument will actually look at different particles and say if it's a single event, a double event, or a triple, it looks like triplet. So if there were 3 events there. I'm going to change this to a linear scale. And I'm also going to change this specific options to be so that we can see a little bit more clearly where we are on the scale here. Now I'm going to introduce just some rectangular gates around these populations. And I'm going to name this processed. I'm going to change the color here because yellow can be hard to see. Single. Double. And then triple. Now, I've already created these gates, and so it looks like they are not aligned with what I was thinking for my original hierarchy. I wanted them to be children of this processed gate, but I didn't quite make them like that. You don't have to delete them, start over. You can actually just drag and drop and put them into the associated or under the desired parent gate, and then that can adjust, and now you can just keep working forward. Now these are actually children of that processed gate. Once I have these gates in place, I am going to insert a data grid. So another way that we can insert images is not just through that picture plot, but we can also do this through the data grid. Here it will give us initially all the parameters and all the information by a cell by cell basis, but not to worry, you don't have to keep looking at all of those. You can choose which parameters to display. So I'm only going to use the ones checked below, and I'm just going to do the bright field here. Now we're going to see every cell event, but like we have this option to see what is processed and then the particle count. I'm going to start using that data grid as another tool so that I can look at these cells based on what is processed and then look at what is in the single event, double event, triple event in this data grid. So just like other plots and objects, we can apply gates here. So I'm going to do processed here because I want to see ones that have all of the images here so that when I'm looking through these other gates, I will be able to see what is happening. So I'm also going to be able to select gates to display. So right now I'm not showing any of the gates. I can do only the gates checked below. And then I can start identifying which cells are falling in which population. So the one that is blank here, it's still a processed event, but it's just not in another gate yet here. So that can happen sometimes. Maybe it was processed, but then it didn't have a singlet, doublet, or went somewhere else on the plot. That's okay. So what I'm going to do is I'm going to pull out this first line of information. Now, this is kind of a unique way. This is kind of a Robin way of doing some image analysis. I like to use this because when I pull it out of this data grid, I can set this as a one of my comparison or one of my ideal images to support whatever analysis I've done. So this is my single cell. And then I have this little segment here that says picture. Well, to pull out a picture from the data grid, I can just select it and pull it out. And then when I pull it out, you'll see that I have a single cell. So it looks like it did a really great job applying that single cell. Now we don't have a whole lot of double events or triple events, so I'm actually going to apply those cells, those gates here, just because then it makes it a little bit easier for me to see that I actually have what is expected. And I'm just going to pull this out here. And that's 133. Pull this image out and then correct, right? Another good finding. This definitely looks like a doublet of cells. Do this one more time for my triple cells. Again, just a way to make sure that one, I'm seeing exactly what I'm expecting to see, and that any of the information, I'm kind of like a confirming of what the plot is already telling me, just confirming down on that data. And this is 5437. Bring out that image, and it definitely looks like there are 3 cells there, almost like a little Mickey Mouse. So maybe that would be a blubbing cell. And again, these images are important for different reasons. If I only want to look at single cells, then I want to make sure I'm looking at singlets. If I'm looking at cell-to-cell interactions, then this could be important. If I'm looking for cells that are potentially blebbing or doing other things, then this triplet column or the doublet column could actually be beneficial as well. Being able to look at these images really helps us confirm that what we're seeing is in line with what's being represented on our plots. So I'm going to add double here, triple here. And I'm just going to scoot some things around here to make us a little bit more space. The last thing I'm going to do with this data file here, I'm going to drag it out again. I'm going to do a density plot. And we're going to take a look at our quadrants. We've looked at all of the other, or a few of the other ways that we can gate information. We're going to look at this quadrant gate now. So when we introduce quadrants into our workspace now, after we click the icon for quadrants, you'll see it's a darker gray. That means it's active. And then when we click on the plot, we get a create new quadrants information. So we can choose whether these are part of the hierarchy or not. We can also choose the parent gate. So when I drag this file from the data list, I didn't designate a parent. I would really like this to be under the process population because those are the cells that I'm most interested right now. So I'm going to change that parent gate there. I can define a prefix if needed, and then it will default to using the name for your current X and Y here of those parameters. So if those are okay, we can leave them there. You can also adjust them as needed. Under quadrant properties, you can also assign these so they will be data specific, meaning that I can move them for each data file and that position will be held. I'm going to leave this at the default setting and then just click OK. And now I have, inserted those quadrants and I can move them around, make sure they're in the correct position. I have them as a child of that processed population as well. So one thing that I haven't shown yet is in our data list, you can see that I have blue iterations. Those are considered active, and now I have gray iterations. FCS Express was expecting me to use samples or to use one data file initially. And this is part of our batch processing, how it runs our data through the data analysis strategy. And it starts at a default of just using one data file at a time. If I put data files in unexpected places, then I kind of deactivate these iterations. And how do I resolve that? On the View tab, going back to that data navigator, it's going to tell me what files I'm using and where. So if I expand these little carets here, it's going to tell me what plots that I'm using these on. So anything that's highlighted, it's going to select that plot so you can see those numbers here, right? And it will actually choose them from there too. So if I were looking for, oh my gosh, I only put my data file in one place, This will help me navigate, excuse me, to the plot and then select it so that I can make that adjustment. Another tool that we have, because sometimes we need to look at different data sets or more than one data set, maybe we have some controls and test samples, is we can also use panels. So right now I'm going to add a panel, give it a nice generic name, and I'm going to set this for just 4 samples because I think my first data set has 4, my second one has 3, so I'll be able to work that out too. We can also set stationary controls, and this is super useful because sometimes we just need controls to hold a place to make sure that we're putting our markers or gates in the correct spots. We don't want to, maybe we have FMOs or isotypes that we're using as controls to designate where our gates are going. We can set this as a control, and then FCS Express knows not to move that data file on that plot. So then you can use those controls wherever you needed to establish those gates. Once I click OK and OK again, my iteration list is going to update. So right now I have 4 data files for that first iteration and 4 for the second one. If I needed to add another data file, I can do that, or I can add a blank tube. So you can add a single blank tube or multiple blank tubes. Now, what's kind of throwing the software off right now is that I have adjusted this so that my 4 samples at the top, and then I'm using different data files at the bottom. I'm going to move this data file down here, and now my blank tube is in a different place. If I wanted to reactivate all my iterations, what I could also do is just move this up. So now the 2 data files are being used as expected. Can adjust this so that it has the same kind of data file, because there's a little bit of disconnect because I'm using so many different tools at the same time. So if there's any confusion there, I'm happy to explain that, but this is just kind of a tool that you can use, our panels, if we're using different controls or different data set numbers. I'm going to go ahead and just uncheck that, and then it will turn that off, and then each data file will go back to 1. As you saw a little bit here, I can adjust how data files are ordered in this data list by using the up and down arrow as well. So moving on, we're going to look at some AMNIS data. I'm going to bring in a data file from there. And in this folder, what's important about our AMNIS data is that we also need to have that CIF file. If the CIF file is not there, the images are not going to be brought in with your data. So you have to have the CIF file that is named exactly the same and in the same folder location as your DAF file to access any images here. I did use the FCS file with just the colon or the period here so that you would be able to see both file types. But when we're bringing in the DAF file, there is a designated file type for that. So I'm going to select that and select my file type here and open that data file up. I'm going to just drag it out from the data list. And click OK. And here I'm going to just start inserting some gates as well. Make sure that we're getting cells. Drag that gate out to create that new population where I'm only looking at the cells that I've selected, and it creates that new data plot. And then I'm going to change this to aspect ratios here. Right, I want it closest to 1, so I'm just going to grab this corner here. I'm going to name that gate 1 and click OK. Now for AMNIS data, you also do introduce a data grid to see those images. So once the data grid is introduced, we're going to see all those parameters again. They're all there by default. After that, we can go back and check just the ones that we would like to see. So this sample is really cool because we have a composite channel, we have DRAC5, we have FITSI, and we have Brightfield. So we have multiple different images collected on a cell, and that is always neat to see how different fluorescent channels are showing up. There we go. Make that a little bit bigger, a little bit larger, and then you'll see those images of those cells. Now, I did talk a little bit about different ways to introduce statistics. So I'm going to go ahead and insert a text box here and right click to insert. Oh, select table and then do insert table. I can define the number of columns and rows here, and then I can click on insert. So I'm just going to do two. One of the most pervasive and powerful tools of FCS Express is really just drag and drop. So I can drag this data file, or this plot rather, into a cell and then let go. So this will show me some tokens. And again, tokens are just really cool ways of saying piece of information. A statistic token is anything from the analysis that I'm creating. A keyword token is anything from inside of the data file. So I'm going to click a keyword token first, just so we can explore that. Oh, this is a DAF file, my apologies. In a FCS file, you will have some keywords there that you can introduce. Here, if I drag this, I can also just insert a statistic token. I'm going to do a file name from here, click OK. And then I can introduce statistics as well. So if I wanted to do statistic token and cells and number of events, click OK. I can introduce that. You can also just type here to introduce additional information. That's one way that we can introduce statistical information. Another way is we can insert our spreadsheets. So spreadsheets are great because we have all these new tools that will be available. You'll see when it's highlighted, I have two new tabs. I have a layout tab and I have a formulas tab. We have multiple different formulas, so you can really do that high level statistical analysis. And then we also have many different ways so we can format this spreadsheet, work with our spreadsheet, add conditional formatting. You can also report directly into this. So if I had multiple pages of information and wanted to do a summary spreadsheet at the end of my experiment, I could report into an internal spreadsheet and then quickly and easily make those charts that would be useful to display my data. So drag and drop is exactly the same as it was for the text box. I can drag a plot into my spreadsheet. It will give me the option to select a statistic token or a keyword token. And I can also select multiple pieces of information. So if I wanted to do cells, gate, let's see, cells and one, because those are gates associated with this, I could do number of events. I could do percent of gated cells from plots, and any other statistical information that would be useful, and click OK. And then those will report into that spreadsheet here. Change the shape and bring it down a little bit. All right, moving on to our last file type that we're going to display before we just look at some generalized templates here. I'm going to add that ICE file. Change my file type. For this, I'm going to introduce a histogram so we can check out using a marker really quickly. I'm going to change this to perimeter. And then I'm going to right click and I'm going to magnify the resolution so that I'm just looking at this section in a little bit clearer way and a smaller range of data as well. I'm going to go ahead and insert from the gating tab the marker. And now a marker, as you can see, I've inserted it, but I don't have anything appearing in my gate navigator. It's not a gate by default. So if I want to convert this, I just right click, convert marker to gate. And then I can do perimeter and click OK. Once I have that gate completed, I can insert another plot, and then I could also apply that gate if needed. Here I'm actually going to insert a picture plot because I do want you to see how these gates, or how this marker is going to influence this. So right now, as we get bigger, you'll see we're selecting those bigger objects. As we're smaller here, you'll see that we have the smaller objects, and they're that bright green kind of showing up there. Kind of see that change there. Now I do have some other layouts because this was just more about introducing new different data file types into FCS Express and Workspace and looking at some of the tools that we have some layouts that have already been prepared that are a little bit more advanced. And here we have one that's a colony counting. So this was segmented. So this is CP out data. So it comes from an image. You'll see that we are able to add gates on the images. So you can see there's a gate here. And then this cell is actually the gate, the one cell that's identified here. So we can take a look at what that image looks like. Again, if we're moving any of our gates, everything downstream is also updating as we make any of these movements and changes. So everything is interactive with any of the changes that we make. On this next page, we are looking at a Nexon and just looking at how our cells are changing cell death, right? So this is our gate here that's adjusting that cell death percentage here at the top. So you do have a lot of flexibility to accomplish many different analysis types and strategies in FCS Express. This last one is pretty cool. We've done some formatting with the plots. We have this nice dark background to really make the colors pop and show off in the beginning. This was the Amnes data file we were looking at earlier. We have different images for these different data files, and that's going to bring me into batch and export. So after we've done all the analysis and established these different analysis strategies, what we can do is go to our batch and export tab. Here under Actions and Reports, we can define some of the tools that we are interested in, or some of the exports that we're interested in creating. Here I have export to PowerPoint, I have an export to a PDF, and export to Excel. Now, when we first add these, we do get a window that opens up to let us define some things for this export. We can open presentation for save. After saving, I like to select this whenever this option is available because I like to see that what I've created is consistent with what I'm seeing in FCS Express. We can include a background from master slide. So if you have a logo or something, you can place it on a master slide and it will automatically be on any other page that you are creating in FCS Express. You can choose quality. So you can do the lowest quality, highest, medium. You can do full page or you can crop to content. One of the things that's really nice is if I wanted to do side-by-sides of plots, I could select a plot and then run it to show just that plot or just the side-by-side of that plot for each of my data files. I can do all pages or select current pages. I can choose to select everything to go into its own presentation or the same presentation, depending on how I want to do things. You can also do things like append to an existing data file, which is really great if I'm doing a longitudinal study and I don't want to have to come back and put all these slides back together at the end. I can just select after I've created the first PowerPoint, I can select this, define where that location is, select that PowerPoint, and FCS Express is already automatically going to add new pages to that report. I can also save it to new. When I save to new, I do have to designate the folder that it's going to, and I can also designate tokens. So if I'm sharing this with somebody and I don't want it to be a pain in the neck for them to have to save, I can do awesome things like just tell the software to save it to the desktop folder. That means they won't have to do anything. It will have whatever name I've given it to. It will automatically save to their desktop, and then it makes it easier for them batch exporting out information when they're getting familiar with FCS Express. And click OK. And so each of these actions do have this window where we can define different elements. They're very similar, so I won't walk through that again. With our spreadsheets, so this is actually an external export. So we have the ability to do a column mode or a cell mode for external exports of our Excel sheets. And here you'll see that I have a few different statistics. If I double click on any of these statistics, I do have the ability to define the field. So what that means is I can customize the name. Sometimes with the statistic naming options that we have available, it isn't quite customized enough. If you need to make those adjustments, you can click on field and then specify what this name will be when you report this spreadsheet out. You'll see that we do have multiple statistics reporting externally. If I needed to add an internal spreadsheet, and I wanted it to be the same as whatever I'm exporting as, I can first define how the information is going to be exported into this spreadsheet. But I have to have a spreadsheet already in the layout. I don't have that, so it's going to probably give me a warning. Um, but this is really nice because once you've added this element, you don't have to then recreate all of your statistics. So I'm going to cancel out this window really quickly and add my spreadsheet. There we go. So it's just going to be a blank spreadsheet because I'm going to report into it as like a final step from this workspace. Go back to my page one, go back to those reports. I'm now going to add this new action to do my internal spreadsheet. Select the spreadsheet. It's going to start in my first available row and column, and it's going to write all my labels. I'm going to click OK. Now, because I want the same statistics here, I'm going to right click, copy, and just paste. And then it's updated. I don't have to go back and recreate anything. I've just moved the statistics and I can do the same thing now if I wanted to do a chart or something in FCS Express. It's really simple. I also have that exported. If you don't want to use a certain tool on a certain day, you can also just uncheck it. You don't have to delete it. So that way it's always maintained and you can decide which actions or which items are best for your report. I'm going to add my data list. I only have a single sample here, so I'm just going to get one report. But then I can click on Run. It's going to create these items for me and give me a little bit of a window to let me know where it's at in these process. And then the items will open here. So I've got my spreadsheet. This is the external spreadsheet to Excel. This is just like an Excel sheet, so I can continue working and doing whatever else is necessary for my analysis. Here is my PowerPoint. I forgot to tell it to just do that one page because I added the spreadsheet. But I would have a nice report here. If I had multiple pages and did just one page, I would have all this information reported and compiled out into a nice presentation here. All right. And then finally, let's go ahead and look at that spreadsheet. So the internal spreadsheet is populated with those statistics that we had reported out from the internal spreadsheet. If there was something that I wanted to report as from this, if I went to my insert tab and did a chart, let's do a bar chart here, right? We have mean, we have file name, not very significant because we're only looking at one data file right now. But if there were more events here, really quickly to introduce a bar chart, pie chart, and simply be on your way after creating these items, then you can just report them out individually as well. So I know I have talked a lot and talked very fast. I'm going to go ahead and look in our Zoom window and just see if we have any questions that need to be answered online. Does anyone have any questions for us at this time? All right, I'm not seeing any questions, so let me think of some commonly asked questions. Going back to our iterations. Iterations come up a lot because when you're running batch export, if these are grayed out, it won't run through all of the data files. So some other tricks, if I'm using other data in other spots, what I can do is I can tell certain plots not to change the data files. So then it can conserve all of my plots where I'm using the data. And I don't have to oops, grab the wrong things here. Let me grab three of a kind here. I can tell it not to change this data file. So then at that point, I won't have to worry about things being disconnected. Let me go over here. one at a time. Now, if you do deactivate this option to change the data file, when I load new data, it won't load automatically. There will be some other little caveats there about using the data. But that sometimes can be a solution if I'm just like, maybe I have an overlaid histogram. Oh, that's something that I actually didn't get to show. So let me go ahead and do that. I'm going to throw out my cells here to a histogram. I'm going to change this to my intensity. Right. Not a whole lot of cleanness there, so I'm going to smooth it out just a little bit and see if I can get that to present a little bit better. There we go. Maybe still not the greatest, but that's okay. And then I'm going to add my second data file as an overlay. And then when I add that overlay, you're going to see I have two different colors right now. Again, maybe not the best cleanest presentation on the plot, but we can also adjust colors and how we're showing this. I'm going to go to specific options and do stacked, and then I can see how they're presenting on that cytoplasm, and there may be a better parameter here for them. We go to overlays, so I can change them both at the same time. There we go, some DNA. And then we can see different presentations of those populations on here. Right now we're using mid-bright cyto. So maybe with no gate, it looks a little bit different. Do some adjustments, but you can definitely do overlays and be able to look at your data comparatively, either between different files or different populations. So if I wanted to use the same data file in different populations, I can change a gate that's being applied so I can see differences between different gates as well. So we have a lot of versatility in that aspect as well. We do just have a few more minutes left of our call, but if you do have any questions, please put them into our chat. I'm going to go over the highlights of today. We are directly compatible with multiple file formats. We are able to work between raw data plots and image plots, and the gates are updating as we're moving them. We have great visualization tools and plots and reports. We can also do those high data transformations. So if you're looking at doing Tisne's or Spades, we have that capability. We can do the batch processing to get those reports. We can do overlays. Cell cycle modeling, we saw a little bit of that on our last spread layout there. We have integrated spreadsheets and regression analysis and so much more. We do offer a free 30-day trial to FCS Express, so please visit us at denovosoftware.com. If you have any questions, please reach out to us at support@denovosoftware.com. We do have some additional resources available. I'm going to go ahead and open those now. We have image cytometry application examples. So we have a Tune SitePix example. We did do a technical note and white paper with them. So if you're loading that kind of data, we can walk you through that process. We have multiple different types of data analysis. So if you're not sure, you kind of want to look at some data analysis options before getting into it, we have plenty of application examples. The image compatibility. This is always updating with newer compatibilities that we're developing. So visit this page, make sure that we have the compatibility with your data files. If not, please reach out to us and we can help you get more information or get this you know, work with the vendor to get that compatibility. On our knowledge base page, we do have image cytometry. We have our own little nook for image cytometry. If you have any questions, you want to just see if it's already been asked. If you go into this area, it can expand these. You can see some of the information that we have for image cytometry. And yes, this is recorded and it will be shared, so you'll be able to access this later if you want to test some of the things. Then support, back to support, because it always comes down to support. We're here to support you. We know getting used to new analysis spaces or doing new analysis with like image cytometry can be a little overwhelming when you're first getting in there. We have technical scientists that are here to help you answer those questions. We do virtual training sessions on Zooms or Teams to make sure that we can see your screen, we can understand what you're doing, and help you accomplish the things that you want to accomplish for your analysis. I really appreciate your time and attention today. Thank you so much for joining me on getting started with FCS Express 7 image cytometry. If you have any questions, the time is now. You can go ahead and answer those questions now. More than happy to answer them. All right. I hope you all have a wonderful afternoon. Thank you so much.